science. Essentially math disguised as dinosaurs and outer space to try and seem interesting.
As someone who intensely studied science from high school all the way through college, I can 100% confirm this. That clip is from a late-night show called Last Week Tonight with John Oliver. The show airs Sunday on HBO and has been collecting GLAADs, Peabodys, Emmys ever since 2015.
It's a fact that needs to be spoken. America's prisons are broken. It's a hard truth about incarceration. Prisons are needed for a civilized nation. But mandatory minimums for heroin and crack.
Stack the system against Hispanics and blacks.
Our prison population is bigger than Slovenia.
'Cause we put people in jail instead of treating schizophrenia.
It turns out, in a complete coincidence, we also wrote a book about Mike Pence's rabbit that has also been published. In fact, while his is out tomorrow, ours is released right now. Now, there are a few small differences between the two books. You'll notice right away that our rabbit has a bow tie, so there's that. Also, our story is about Marlon Bundo falling in love with another boy rabbit because our Marlon Bundo is gay, just like the real Marlon Bundo.
The show is so incredible because it takes well-known topics and then gives the audience a deep dive and closer look into the details that they may not know about. The show is so good and it is one of my favorites, and like any other show they take breaks between their seasons, but specifically for this show, when they come back, I'm like...
All right, this is it, people. Let's move. Let's go, 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 go.
Today, I'm going to talk about how this show is just a fancy version of the Central Dogma. And if you don't know anything about the Central Dogma, well...
You're gonna learn today.
The Central Dogma explains how information from DNA gives rise to proteins. It's super important because it tells us how genetic information flows throughout a biological system. Knowing this is how we know that parents will pass down a specific eye color or hair color to their future children. Now, the development of the Central Dogma is credited to James Watson and Francis Crick, even though a major scientist by the name of Rosalind Franklin played a major, huge role in everything that they were doing in their research to come up with this process. It's well known that it took me a while to get comfortable with the things in science, and most of the time I went to class and would take notes, and when the professor would ask a question, I would give wrong answers confidently, like this woman.
Name Popeye's favorite food.
Chicken! Oh my God! Don't you have to be stupid somewhere else?
Not until 4.
But the Central Dogma was something that I was able to grasp pretty quickly because it's something that you can physically see and you can metaphorically get a sense of. The Central Dogma says that DNA goes to RNA and RNA turns to proteins. And this entire thing happens in 2 steps. You can also call it transcription and translation, which is just breaking down the 2 steps that are going to occur. In biology, sometimes knowing which step comes first or what part of a process is going to go first can be pretty tricky. But for transcription and translation, that one is pretty easy to remember. Transcription has a C in it, whereas translation has an L in it. C comes before L in the alphabet, therefore transcription comes before translation in the Central Dogma process. Transcription is just what it sounds like. It's when information is going from one written form to another, whereas translation is taking that middle written form and is forming it into a final product, which is going to end up as proteins. And proteins are important in cells because it helps with everything that we do and goes into gene expression. This is where I started seeing parallels between this scientific process and John Oliver's amazing show. The first step in his show was doing the same kind of thing where he's collecting information. He's doing it with interviews, he's doing it with articles from local and national sources. His interns are running all over the place to collect him the things that he needs to create these stories.
Stupid shows like ours lean heavily on local papers. In fact, whenever this show is mistakenly called journalism, it is a slap in the face to the actual journalists whose work we rely on. I'll give you just one example. 2 years ago, we ran a piece on state lotteries, and a not insignificant portion of it was built on the work of Harry Esteve, a reporter at The Oregonian. Here is a clip we used of him talking about his series on camera. Here I am quoting one of his Oregonian stories directly, and here I am doing it again. You probably didn't notice it at the time because you were too fixated on my bold choice of shiny grey tie with checkered dress shirts. The tie says Mafia Funeral, The shirt says high school debate tournament, and the face says, I am not confident enough to carry this look off.
This is also where he's going to do like one-on-one talks with people and do experiments to get first-hand experience of the situations that he's talking about.
Look at Robert Tilton. If you ever send him a donation, you cannot imagine what happens. And luckily, you don't have to imagine because, and we should probably come clean here, we have been involved in a correspondence with Robert Tilton's church for the last 7 months to try and find out what he tells people. So settle in, because this gets incredible.
This step is also super important because it's the phase where he would be talking to lawyers to make sure that he doesn't get sued for publicly criticizing brands, companies, and public figures.
And I know that after tonight, Murray will probably sue us again. Even though everything I've said has been rigorously vetted by our lawyers, who, may I add, are getting very tired with us. Because when we reached out to Murray last week to review the details that we plan to include in this piece, he wrote a letter back, not addressed directly to us, but to our bosses at AT&T and HBO, saying we were falsely vilifying him, launching scurrilous attacks, and arguing that none of his lawsuits are slap suits, which they absolutely are.
In the cell, during the transcription process, this is when your DNA is being turned to RNA, specifically mRNA, which the M stands for messenger. It's important to note that DNA and RNA are pretty similar since they are both nucleic acids. Nucleic acids are just nucleotides that have been linked together. Nucleotides are molecules that consist of sugar, phosphate, and a nitrogen base. Even though they're both nucleic acids, they're going to have some major differences. DNA is double-stranded, whereas RNA is single-stranded. DNA is deoxyribonucleic acid, where RNA is just ribonucleic acid. DNA typically has a long lifespan, where RNA has a pretty short lifespan. Basically, DNA is Beyonce and RNA is Michelle. The process starts with RNA polymerase. And RNA polymerase is the enzyme that is responsible for taking the DNA and copying it into an RNA template. RNA polymerase goes to the DNA and then attaches complementary RNA bases to the DNA. C is going to be paired with G, G is going to be paired with C, T is going to be paired with A, and A is going to be paired with U. I know, that's a bigger plot twist than learning that Mariah Carey can't actually sing.
Make my wish come true. I'm just gonna keep on waiting.
Let's take a closer look at this animation. DNA is the blue open strands on opposite sides. Notice that the DNA is open and that there are tiny bases attaching to one side, creating the red strand. The red strand is the mRNA. At this point, the mRNA will venture out of the nucleus into the cytoplasm and attach to some ribosomes. This makes sense because ribosomes make proteins, and proteins are responsible for gene expression. And after we are out of the nucleus and officially in the cytoplasm, we can start translation. Now, before we look at translation in this cell, let's go back to John Oliver because he describes it perfectly in his show.
What happens at the Supreme Court is way too important not to pay attention to. And yet, the ban renders any coverage basically unwatchable. But I think we may have a solution. And it's learning a lesson from this. Think about it. If someone made you just listen to the audio of that, you would punch them repeatedly in the face. But the visual makes it irresistible. Why? Because a cat's paws are doing things you wouldn't expect them to do. And that is why, this week, we spent an incredible amount of time and an almost immoral amount of resources to produce an entire Supreme Court featuring real animals with fake paws.
As far as searching the beard is concerned, why can't the prison just give the inmate a comb? You could develop whatever kind of comb you want and say, comb your beard. And if there's anything in there, if there's a SIM card in there or a revolver or anything else you think can be hidden in a half-inch beard, tiny revolver, it'll fall out.
Those dry constitutional arguments are now must-see television.
For last week tonight, the translation step is taking all the information he collected and putting it into a way that is understandable for viewers, but also enjoyable and entertaining to watch. This can be done in the form of jokes or skits or elaborate pieces that he does to really drive his point home.
Let me try to explain why to the Miss America organisation through the only medium it seems to value: 20-second conversations with women in evening dresses and sashes. Please join me. this last week tonight, I'm proud to say, as of now, we are the world's largest provider of scholarships for women, because tonight, 400 million $1 scholarships will be made available to the winner, of which she may choose just one. Look, there are certainly better voices than mine to speak on behalf of Puerto Rico. Luckily, there is someone with Puerto Rican parents and an amazing voice, and he is here tonight. Please welcome the great Lin-Manuel Miranda.
Yeah, my family's from Puerto Rico, the tropical destination, where you can spend your Washingtons, the spot where you vacation. A commonwealth with not a lot of wealth, a not quite nation, $70 billion topic of conversation. Hoping to God John Oliver's comical dissertation resonates with the Congress that got us in this situation.
For the cell, translation is pretty similar. It's taking information and putting it in a way that the cell's going to understand. It's going from nucleic acid to amino acids. And this is important because amino acids are the things that make up proteins. This seemingly simple process actually has a few key players and steps that aren't ever really recognized. The biggest player in this process is tRNA, and T stands for transfer. There are a bunch of tRNA molecules floating all throughout the cytoplasm, and they are carrying the amino acids that are going to make up the proteins. Basically, the tRNA reads a codon and brings in an anticodon. A codon is created with 3 bases, and an anticodon is the complementary base pairs. For example, if a codon is AUG, the anticodon would be UAC. These codons code for different amino acids and give instructions of when to start and when to stop translation. There are tons of mRNA codon charts that you can find online that'll tell you what amino acid you're going to get with different bases combined. It's important to know, though, that you can have different bases combined and still get the same amino acid. For example, both codons UCU and AUG code for the amino acid of serine. The process of adding anticodons to codons keeps continuing until the amino acids are linked together by peptide bonds, and then these keep growing into an extremely long chain that is creating the different proteins. And this continues all the way until there is a stop codon that says all of the process can come to a halt because it is done. This stop codon doesn't have an amino acid that's attached to it. It literally just gives instructions to stop translation. At this point, both transcription and translation is done, and the protein will go through different modifications, edits, and folding to prepare to do its job.
I am the new queen of daytime talk!
While it's important to note that the central dogma is widely accepted and is what scientists think of as the process of transcription and translation, explaining DNA going to RNA and RNA going to proteins, it's not always the case for every organism or virus that we see on Earth. There are 3 main examples that you should know that conflict with the DNA to RNA, RNA to protein process. The first one is reverse transcription. Basically, this is the one where you've got RNA and it's turning into DNA, specifically a type of DNA that is called cDNA, where C stands for complementary. Reverse transcription can be seen in viruses like HIV, and they're able to take the RNA and go into DNA and then can replicate that DNA, spreading and infecting a host. The second example is known as non-coding RNA, also known as ncRNA. And this RNA does not need to go under the process to become a protein because it functions perfectly without doing that. And examples of that are tRNA, which made all of the huge process for translation happen, and also rRNA, which is for ribosomal RNA. The third and last example are RNA viruses. RNA viruses start with RNA, the RNA is replicated, and then it goes straight into becoming a protein. There's no DNA that is in the process. There are plenty of examples of RNA viruses, going from the flu, SARS, measles, even the coronavirus. This just proves that in science, knowing how something will work is just as important as knowing how something won't work. Anyway, that is it for this video. Thank you for watching. If you like what you see or want to see science concepts talked about in a way that is with pop cultures, TV shows, movies, anything of that nature, then go ahead and click the subscribe button, because I make videos every other week. If you would like me to talk about a specific science concept, be sure to leave it in a comment down below and I will check it out. Until next time.
congratulations on being the first subject of our Great Minds series. Be honest, is this the single greatest honour you have ever received?
Yes.
When you say that, are you being sarcastic?
Yes.
I thought so.